all. This is Dr. Mobeen Sayed from drbeen.com. Welcome to a great talk today. Today with us, we have Dr. Jacob Glanville, such a decorated scientist, such a glowing resume. I would love to just read that all to you and have you see what a wonderful researcher, what a wonderful chief executive he is of his company as well, and his contributions to us all. So I'm going to read a little bit of his bio to you, and then I would request Dr. Glanville to talk a little more about himself and his company. So Dr. Glanville here, he's an entrepreneur, inventor, and a computational immuno-engineer. So his company has been working on a universal flu vaccine and an antibody treatment for COVID-19. And check this out. He is in the Netflix documentary Pandemic. I hope that you know Pandemic. And then he is also part of the Stanford University Scientific Advisory Committee member for the Sean Parker Center for Allergy and Asthma Research. He is a scientific advisory board member of the University of San Francisco Biotechnology program. He is a repeat Gates Foundation, Stanford University Computational and System Immunology grant recipient while he was doing his PhD, or while he was a PhD candidate at Mark Davis at Stanford. He is a recipient of Pfizer Achievement Award 2010 while principal scientist at Pfizer. So welcome, Dr. Glanville.
Dr. Bean, thank you for having me on.
This is a pleasure and an honor to have a person like you with us. Thank you very much for coming over. Can you please tell us a little bit about your own background and your company? How did this whole thing come together?
Sure. So I founded my company in 2012. My background is a combination of a couple different specialties. I focused on immunology. I found it always fascinating. I'm an asthmatic, and I grew up in a Zutawil Mayan village in Guatemala. So I was always interested in medical interventions and how immunology seemed to be at the heart of almost every aspect of medicine that I looked at. I was also, just as a hobbyist, interested in computers and math. And I was delighted to find the interplay between these two disciplines, because the immune system inherently uses combinatorial methods to be able to compete with viruses and pathogens that are able to evolve faster than we are. And we defend ourselves by creating these complex arrays of antibodies and T cells and cytokine interaction pleiotropic networks. And that always fascinated me. And large amounts of data, elegant algorithms, and math are one of the ways to attack and understand how the immune system works, or why it's failing in the case of vaccines that seem to miss the important parts of a virus that don't change every year, or why our immune systems sometimes cause autism or let cancer sneak through. I also am interested in bioengineering. So this is the roll up your sleeves, blue collar work of finding ways to manipulate antibodies and other proteins produced by life and turn them into medicines. This is a pretty powerful technique. This is really the medicine of choice elected by evolutionary forces are the antibodies that pump through our veins, and they have been used by all jawed vertebrates. And there's been no organism we've ever seen that's decided, no, we don't need antibodies anymore. That's just a consistent feature of life forms. And the biotechnology industry has learned how to tap into this powerful new class of medicines, these biologic antibody therapeutics. And so that's what my company does, is we use machine learning, computational guided methods, high throughput data acquisition to help speed up and improve our ability to engineer really good antibodies, to take as much learning as we can from how nature produces antibodies, but then engineer them further to improve certain properties that we may need to take beyond nature to make them really effective medicines, like potency, concentration, thermostability, and so forth.
Got it. Thank you very much. And tell me a little bit about the COVID-19. So you are working on a universal flu vaccine, correct?
That's right. Yeah.
So if you can tell me about both of them, first your work on the flu vaccine, and then if you can talk about the COVID-19 as well. We are, of course, at this time, very much interested in the COVID-19 work.
So the Netflix documentary was following us in 2019. My lead scientist, Sarah Ives, myself, and many of my other scientists, as we were working on a broad spectrum or a universal vaccine that could treat all the versions of flu. So the current versions of flu shots, they if you're lucky, they will cause you to produce antibodies against that year. So they protect you against that season. But the flu mutates every year. So you need a new flu shot every year because your old flu shots become obsolete. The thing you're producing an immune response to is no longer what the new virus looks like. We were trying to fix that to create a single vaccine that would protect against all versions of the flu. And the way we were doing that is we were trying to teach the immune system to recognize shared spots on the surface of the virus that the virus is not able to mutate year after year. These spots do exist. We've actually found them on flu. Scientists have found them on HIV. Many viruses have these Achilles heels that cannot change from year to year. The fascinating thing about those is that if you can target them, you have created a universal vaccine for that class of virus. The challenge and the mystery is that why is it if that site doesn't change every year, why does our immune system so often miss to target that site? And we had a technology to successfully overcome that. That work ended up winning a Gates Foundation grant, and we're currently testing it in ferrets and pigs with a live challenge study supported by the Gates Foundation to move that work forward into humans. The COVID work, we're actually not working on a vaccine. There are other groups that are doing it, and I'm glad they are, but we are working on an antibody therapeutic. So the difference between these two is that with a vaccine, you're injecting little pieces of the virus into a patient, and then that patient's own immune system will spend 6 or 8 weeks learning how to make good antibodies that target those pieces of the virus so that if they ever got infected, those antibodies would then latch onto the virus. That's they're inexpensive, they're powerful technology, but they take time to work. The challenge is that if you have someone who's already sick, you can't give them a vaccine because they don't have 8 weeks to wait. They need a medicine now. And so for that group of people, so the people that are already sick, you need a therapy. And what we're doing in my lab for the COVID-19 is we've been building an antibody therapy. So there, instead of injecting pieces of the virus like a vaccine, we're just directly injecting a set of very potent antibodies. And those antibodies flood in their system. And what they do is they go and they stick to these spikes on the outside of the virus capsid, and they block those spikes. So the spikes are no longer able to stab your cells and inject their nefarious genetic payload and infect you. We right now know that our antibody is very potent in its ability to protect hamsters from the COVID-19 causing SARS-CoV-2 virus, both prophylactically or preventatively as well as therapeutically. It's in manufacturing with a company called Millipore and another company called SwiftScale, and we're anticipating running clinical trials towards the end of the year.
Got it. So this is excellent. And I had this question, and this question has been asked by many other folks as well, and you touched upon that. The antibodies versus the vaccines. Is this antibody now can be given for prophylaxis as well?
Sure, yeah. So we've engineered, so antibodies can be given both for therapeutic. So you have someone who's already sick, usually an IV, but you can also do an injection if it's very potent. You give them the antibody and then it will sort of act as though their own body had figured out how to produce a good antibody. It will block up the virus and protect them. You can also choose to give an antibody to someone who is not yet sick. So we did that with hamsters. We gave them the antibody first, and then afterwards we exposed them to the novel coronavirus. And when I say we there, I mean the Department of Defense, US AMRID, they did a partnership with us. So they ran those studies because they need the special biosafety level 4 facilities with the spacesuits where scientists can work with the coronavirus safely. The reason we ran that experiment is that is something you may want to do with people. So vaccines are great, but they don't work particularly well on the elderly or the immunocompromised. And they also have the problem of taking 8 weeks to work, and they don't always work. So for those people, those unfortunately, the elderly and the immunocompromised are at very high risk of complications with this virus. It would be nice for them to have a preventative. And so our antibody, we could potentially inject into the elderly and the immunocompromised, and then they don't have to be dependent on a vaccine, their own immune system, to produce the antibodies because we've made it for them. We've also engineered our antibody. We've added in these mutations that cause the half-life of the antibody to be significantly extended. So a single injection should be able to last somewhere between 8 and 10 weeks. And after that, they would need to get another injection.
Got it. So it seems like you read my mind. This was my next question, that normally antibodies break down earlier. So 8 to 6 weeks. So would the cost be something that would be affordable for someone to continue to have maybe 3 or 4 doses in a year?
Yeah, so that's cost is a big part of how we're doing things differently. You may have heard about the remdesivir release. That's a medicine that reduces the hospital stays from 15 days maybe to 11 days, so it helps a bit. But they're planning to charge over $3,000 for a patient, and I think that's just far too much. That's certainly too much in the village I grew up in Guatemala, which means most of the world. That's too much, but even that's too much in the United States. There's a lot of people that don't have good medical care, and we need something that everybody can take. Certainly, as you said, if they need to come back and take it multiple times a year. So with our therapy, it is usually the case that antibodies are quite expensive therapies. They typically charge somewhere between $3,000 and $9,000 a dose to receive an antibody therapy. The cost of goods of an antibody therapy is nowhere near that. The cost of goods is about $100 for a dose of an antibody therapy, for a gram. The rest of what you're paying for is corporate profit, and it's really the ruthless calculation that the corporation is estimating how much money would I be saving the insurance agencies by offering a therapy that would reduce the amount of time they're having to spend other money for other reasons on that patient, and then they half it or take a fraction of that, and then that's what they charge to the insurance agency. So we don't want to think that way. Instead, we want to say, okay, what was the cost of goods? Let's say it's about $100. What's our total business operational cost and distribution and so forth? That may be another $100. And then we'd like to put a profit. We do want a profit on that. I'll tell you, it is very annoying to try to run a business through grants. I don't need to be greedy, but some money is the best way for me to make new medicine.
Absolutely.
So, but again, the profit could be like another $100. So we could be charging $300. And I don't know the exact price. Maybe it's $300, maybe it's $400, but less than a cost of an iPhone for a life-saving therapy. That's something that someone could take multiple times a year. That's also a medicine that anyone in the world could afford. And that's what we're aiming to produce right now. And part of the reason I'm able to do that is, first, I don't have venture capitalists. I never took on venture funding, so I'm not obligated by fiduciary duty to maximize the charge. The other advantage is that I engineered these medicines incubated in my own company, and my company has been profitable since founding in 2012. So I don't have 10 years of sunk cost. A lot of companies say, well, we've spent so many years on research and development, we need to recover those costs by charging a lot for a medicine when we finally make it. But I'm kind of lucky. I made this medicine, I don't owe things. And the final advantage is that a lot of pharmas, they have 30, 50, 100,000 employees. And when you have that many employees, your costs are huge. You need to create blockbusters as just to be able to have the economic metabolism be satisfied of such a large organization. Whereas right now, I have 35 employees. We'll grow a little bit, but the goal is to be strategically small. And I always see the future of my company to make keep the company as small as possible, to outsource certain tasks to contract research organizations whenever possible, and take advantage of the new generation ecology of distributed services, rather than building the biggest company I possibly can. Like, I think sometimes people just want to build a pyramid so they can take a picture of it and show it to their moms and show off. But honestly, we could keep companies smaller and make more affordable medicine for the world. And I think that's a better use of our time. That's the photo I'd rather send my mom.
I love it. So thank you very much. And I think that, as you said, for life-saving medicine, $300-400 per dose or per injection is not much. I know that in some areas, some countries, it is going to be much, but still we are talking about a life-saving therapy. So tell me one more thing. So this is on the prophylaxis side. When a person is now ill with COVID-19, and the stages, as we've been seeing, that there could be mild, moderate, severe, critical, and even then afterwards, the long haulers, a new emerging state where people have the symptoms, not knowing that is it the immune system that has gone in an imbalance, or it is the virus that is still sitting in there somewhere. At what stage this therapy can also be used other than prophylaxis?
Sure. So what we know from Ebola, rabies, other diseases for which antibody therapies have been very effective, and from what we know from remdesivir and just from understanding the basic theory of how a neutralizing antibody works, the general idea is the earlier you can give it to the patient, the better the benefit. Because what this antibody is doing is it's blocking the virus, so it's neutralized. We know in the hamsters, we gave the antibody at 15 mg per kg, which is a reasonable dose, to hamsters that were already infected. And within 2 days, those hamsters had 97% reduction of virus in the lungs, and they had a resolution of the majority of lung pathology. That tells us that the antibody can do its job, but it's going to do a really good job if its main goal is to stop the virus from spreading. If the virus has already been spreading for a long time, it's starting to cause a whole bunch of other tissue problems. It's causing the immune system to hyperreact, create something called a cytokine storm. And that means that even at that point, if we do block the virus, there may be runaway reactions happening in the body that are causing other problems. And so what we anticipate, again, we have not run the clinical studies yet, but we anticipate what we're going to see is that the earlier you get it, the more it avoids you deteriorating into these more complicated states. By the time you're already a critical patient, we still think an antibody can help, but there we think the steroidal effect, the steroids and some other medicines may actually be more important at that point because a lot of the damage is already done. So we anticipate to see the majority of the benefit early. The way we're constructing our clinical studies is that as soon as the patients come in, they're already serious enough that they showed up at the hospital. Ideally, getting them earlier would be even better. But by the time they've arrived at a hospital, but before they've deteriorated any further, that's when you'd want to give them the IV to be able to treat them. Now, with respect to the long haulers, it's unclear to me whether it's going to help at all, because the long haul pathology, I don't know, is that due to any residual virus or is that due to clots and tissue damage and a series of other problems where if the virus isn't present, this drug wouldn't do any good. It's this drug's job is to go stop the virus from spreading in your body. It'll do that really well, but the earlier you can take it, the better.
Totally makes sense. So the takeaway here is that the therapy itself is useful when the virus is present.
Yes.
Because the therapy is going to go and neutralize the virus itself. And if the virus has done the damage and has gone down to a very lower level, and now it is the immune system or the body tissue damage that is now just causing the remaining issues, then the therapy may not be very valuable. So that means prophylaxis, that means early use of this therapy should be very, very important.
That's right. I think also, severe, serious, also very good. By the time, seems like there's an inflection point when intensive is described. By the time you get to the point that the person needs high pressure oxygen or intubation or they've undergone a major cytokine storm, they have a new whole class of problem. An antibody like this could still help, but at that point they also need steroids and they need immunosuppressants and oxygen. And it's sort of a different type of disease that's being dealt with at that point. Up until that point, we can definitely help them with an antibody. One of the other things that we've done with this antibody is that we've engineered what are called the Genentech LALA mutations. These are a couple of mutations in the FC, like the antibody is shaped like a Y. At the base of it, that's the handle of the immune system. The top arms, it grabs onto the virus. The base is where the immune system grabs onto the antibody. And it seems like with this virus, there's this problem of antibody-dependent enhancement. This is a little bit complex and incompletely understood in the context of this virus. But what it means is that sometimes antibodies will grab the virus, which is helpful, but they may also make things worse by enabling the virus to be internalized into immune cells, infecting immune cells, or causing the immune cells to freak out and release a lot more cytokines. We have created our antibody to be more safe by removing the effector function so that the handle basically cannot be grabbed onto by the immune system. And that means our antibody is sort of a stealth fighter that sneaks in, it blocks up the virus, but it doesn't freak out the immune system. And we think that's going to neutralize the virus without causing additional inflammation and tissue pathology. And that certainly appears to be the case in the hamsters.
I love this. This is a stroke of genius. I love this, that the immune system itself is not going to go crazy while the antibodies are coming up and they are mopping up the virus itself. So I want to ask you one more question. That is very interesting for me, and many folks have been asking me this. And so let's acknowledge that there are folks who are not very much fan of vaccines.
Yeah.
Now, that is a wrong thing or a right thing. Let's not go there, but let's just acknowledge that there are folks who would not like the vaccines for multiple reasons. Can they take advantage of a therapy like this, passive vaccination or passive immunization or the antibodies, and do they have similar issues that vaccines may have, or these are safer, better therapies?
So there's going to be a range, but in general, here's the big difference. There in the United States, Dr. Fauci had told us that there's about 20% of Americans who say they will never take a vaccine because they're vaccine skeptic. And then there's another 30% of up to 30% of Americans, so that's 50% in total, either won't take it or are concerned about taking a new vaccine, and they're worried it may be rushed. The problem with that is that that's suddenly enough of the whole population that won't take a vaccine that we may not be able to establish herd immunity if we have an effective vaccine. And it looks like these vaccines are working. They're producing good antibodies so far. The nice thing about an antibody therapy, it's not, I'm not going to convince everyone. There's going to be some people who say, I never want to take medicine, and that is what it is. But it's a very different thing to ask someone to give themselves, when they're feeling healthy, a preventative, than when they're in a hospital and they're feeling sick and they're scared to take a medicine to get better. And most people who would not take a vaccine when they're healthy are going to tell the doctor, give me anything it takes if it's going to make me better, when they're scared for their life when they're sick. And so I think that is helpful. My other attitude here, first off, I think we want both vaccines and therapeutics, but part of the reason I worked on a therapeutic was that my feeling was, because of the nature of this virus, that 90% of people don't get that sick, and most people who get sick produce antibodies. But then the 10% of people who do get sick get extremely sick. They're at high risk at hospitals. They have long recoveries and have elevated risk of death. My feeling was it was actually more important to have a therapy than to have a vaccine. And we want both. But imagine that if we have a vaccine, we haven't really solved the problem, because we're going to spend all of 2021 vaccinating people. A bunch of people won't take it. We're going to continue to have this crisis. Whereas if we have a therapy, then 90% of people can get sick. Half of them won't even know. Most of those people will develop antibodies after and then sort of enjoy natural immunity. The people who get sick can go to a hospital and they're no longer scared, because they receive, as provided it's effective, they can receive a therapy, they get better, and they go home quickly, which means we don't have the additional terrifying burden on the hospital settings, and that could help life resume. Now, ideally, you have both. You have people being vaccinated at an accelerated pace, and you have the safety net of a therapy at the hospitals. And I think that's really going to help out. And I think even most vaccine skeptics, or people who are concerned about taking a vaccine, would willingly take that antibody when they're at the hospital. If people choose not to take a medicine at the hospital, I mean, ultimately, my feeling is that's their right. It's their body. But I feel like most people who would be skeptical of taking a vaccine when they're healthy will change their mind when they're worried about their life in a hospital.
Makes sense. So one more question, and this is something that I've been thinking about a lot. So a few months ago, many companies had come out. I think even you had an announcement as well that you have the antibodies. Israel came out and they said, we have the monoclonal antibody. There are many other countries, many other labs that came out. Why don't we have an antibody available till now?
Yeah, so what we announced and what other groups announced is that, I'll speak for myself and then I'll say where everyone else is in relation. We proved that our antibodies are able to neutralize the virus. We sent it out to 5 labs in 3 nations, and they all confirmed that our antibody, when you mix it with the virus in human cells in a petri dish, it blocks the virus from infecting human cells. Then we sent our antibody out to 2 labs that confirmed that it protects hamsters so that they don't get sick when the antibody is present. That shows that this could be a good drug. You still have challenges ahead. So your next steps is that you need to manufacture the drug at a high quality standard. This is called CMC, GMP production. And that's where we are right now. We're working with a major antibody manufacturing group called Millipore. And we're also working with a second technology called SwiftScale. So we're producing our antibody in 2 different ways. We're in the middle of that manufacturing process. You also have to complete something called safety and tox. And this is a set of tests to make sure that your antibody is going to be safe when you administer it to people. That's ongoing with us right now. We're working with Charles River Laboratories, and they've actually subsidized that entire part, which we're very thankful for. So they're paying for our safety and tox for our therapeutic program. What they do there is they give a high dose of the drug to rats to make sure it's safe at a high dose. They also take the drug and they put it on human tissue slices from human cadavers to make sure the drug isn't staining, accidentally going to stain your liver or cause any body problems before we give it to real people. Once you've completed that, then you have to go into clinical trials. So you need to give it to people in hospitals, and we have statisticians and careful rules to make sure that we can say, yes, definitely it's the case that the people who received the drug recovered faster, their oxygen got better, they went home faster, they were less likely to die. And that's really what proves that a medicine is worthy of being approved by the FDA. When we're talking to the FDA the whole time, there's a process called an IND that we're going through, and they've been extremely responsive. So we are going through this process, so are all the other companies, and we're all on a race, but that process does take many months to manufacture the material and to go through the safety and tox and complete the clinical trials.
Got it. So any expected time that you would be able to start your trials or have the vaccine available?
So here's where the world is. With respect to antibody therapies, there are a couple other companies that I think are going to reach market a little faster than us. I think our medicine will be more potent and safer and have a better half-life, but they will reach first, which is good because that also shows I want to watch their studies with great interest, because if theirs works, mine should work at least as well and potentially better. They should arrive around December, maybe January. Mine will show up a little later. The vaccines, I know we have 3 that are in the lead right now. There are, I think, almost 180 vaccines under development. Those look pretty good, and I think those will also be available at the end of the year. So I think it's reasonable to guess that we will have effective vaccines and effective antibody therapies at the end of the year and into early next year of 2021.
Got it. So 2 more questions. I'm going to put both of questions together. The question is, are there going to be complications or side effects? So of course, you're going to be testing and you're going to look at the side effects. But once it is out in production, what are the general side effects of a vaccine? And the reason I'm asking this question, one, there are questions that are being asked in the comments here. The second is, can we have such therapies over the counter?
Okay, so a couple questions. Yeah, first off, side effects. And the side effects I'll answer for vaccines and for monoclonal therapeutics. So for vaccines, there are relatively common side effects. And I will say that the 3 vaccines I've looked at for coronavirus, they all seem like they have a lot of these side effects. And those are that when you receive the vaccine, most of the people who receive these vaccines, for a day or 2 after they received the vaccines, they had some kind of seemed as though they had a cold. So they were having pain at the site of injection, body aches, a fever, general feeling of not feeling so good. Those are probably worth it compared to the risk of getting the corona. You do not want to get this coronavirus. It has a whole bunch of complications and you're on a long road to recovery. It is not the flu. So I would prefer that to the alternative, but those are side effects. It is possible to have some more extreme side effects for vaccines, and that needs to be monitored carefully. One of them is the risk of antibody-dependent enhancement, and that is we need to make sure that these vaccines, the people who are vaccinated, when we watch the phase 3 trial, we want to know, are the people who are vaccinated less likely to get COVID? And that's what we're expecting, but there is a possibility that having the wrong antibodies in your body is actually worse than having no antibodies at all. It could exaggerate the disease. And this is something that has been observed in other coronaviruses for vaccines produced for cats, that we found that there was an antibody-dependent enhancement effect. This has also been observed in dengue, but the wrong antibodies are worse than no antibodies. The data I'm looking at so far, to me, says that's unlikely to be a major problem, but it is a risk, and it's something immunologists are looking carefully at. There are also, some people who receive vaccines, it's possible to have a seizure and have other effects. These are relatively rare, but these are things to be monitored for to make sure that the medicine is safe, and we're going to find that out in the phase 3. People worry about autism and things like that. That is not substantiated in vaccines. I've never seen a case of that. In extremely rare cases, it is possible to get some autoimmunity that is possibly triggered, and that's something that's going to be watched for, but it is much less common than I think people worry about. Now, with respect to antibody therapies, antibodies are produced in your body, so they usually are very safe. We infuse antibodies all the time. There's actually a product called IVIG, which is just a mix of a bunch of blood donors' antibodies that get infused into people all over the United States all the time and the world. That said, you can have a reaction infusion at the infusion site. So if you have an IV, sometimes there'll be like an inflammation around the site of infusion. When we check for that, it's possible to have sort of an allergic reaction or that your body begins to reject the antibody, and we'd be checking for those things as well. But historically, antibodies are very safe, particularly if they're pointed at a virus and not at one of your own tissues. So that's part of the reason why they've been promoted super quickly, is that they're very safe and they've had a history of being very effective against viruses. The other thing with an antibody is it lasts for, in my case, 8 to 10 weeks, and then it would just go away. So it washes out. It has a finite lifetime in your body before it goes away, and that's an additional safety feature.
Got it. So now imagine, let's indulge for a second that it is safe enough. Is it possible that these antibody therapies could be over the
Right.
counter?
So one of the things that we have been trying to engineer with our antibody is a new route of delivery called sub-Q or subcutaneous. Normally, antibodies are delivered through an IV bag, so you'd have to go somewhere where a nurse or a doctor or a phlebotomist training person can go and set you up with an IV bag so that you can be infused with the antibody. That limits the people who can receive an antibody. We are looking into whether we can concentrate our antibody enough so that it can be delivered in a syringe. And the power of this is that it no longer needs to go into a vein, and you no longer need a specialist that can plug an IV bag into a vein. You can inject it into the subcutaneous, the area under the skin, typically on the belly. That would be really powerful because that means we could have someone go with a suitcase full of syringes and just go over to an old folks home or a Navy boat and inject everyone, and they'd all be able to have the prophylactic protection. The challenge of subcutaneous injection is you need to have a very potent antibody, which we have. You need to be able to concentrate it quite a bit. And we worked on this when we engineered our antibodies. We heated the population of billions of versions of the antibody up to 72 Celsius, so quite warm, so that anything that wasn't stable wouldn't survive. And we just picked the antibodies that survived that strong selection pressure. And that means that these antibodies are very stable and you can concentrate them without them aggregating and turning into basically scrambled eggs. But we're still running those experiments to confirm the concentration we can achieve. That is definitely an item of interest because you still need someone who's like authorized to use a syringe, but that gets you closer to being able to do a out of hospital setting. It is maybe possible that we could do, in order for it to be over the counter, it would need to be an injectable device that was sort of foolproof where someone couldn't hurt themselves. These things are possible. We know that diabetics, for instance, and there are other groups that have puncture-based assays that can be performed. There's also patches and a couple other things that could be considered. But I think at this point, the short term, we're going to have IV bags for hospitals, and we're aiming to do subcutaneous for out-of-hospital delivery of the therapeutic.
Got it. Thank you very much. So I have one more question from me. Then we'll look at some of the questions from here if you have time from the cool beans, our viewers. So one question that I have, and we've been talking about the cool beans here, we've been talking about it for some time, and that is the mutation. If there is a mutation in the virus, can that defeat a neutralizing antibody that is connecting at a specific epitope, and now that epitope has changed? Virus is still active while the antibody cannot do anything. Is that possible? And if it is possible, how do you fix that?
Yeah, so that is possible. Our antibody binds the receptor binding domain. So this is the tip of the spike on the virus. This is the part of the virus that goes and binds the ACE2 receptor. Our antibody goes and it blocks that interaction. It's possible that if the virus was able to create a mutation that made our antibody no longer bound, then it could escape. Those viruses would escape our antibody. The challenge is that our antibody binds right on top of where the ACE2 binds. So any mutation that makes, most of the mutations it would make to escape our antibody would also cause it to no longer bind the human cell well either. But it's not out of the question that it could create just the perfect, very annoying amino acid change to cause an escape mutant. That's something we're looking at carefully with BARDA. One solution, which is something that's been recommended to us, is to create two antibodies. So you have two antibodies that both neutralize, and it's like an antiviral cocktail where any mutation might escape one of the antibodies but not the other one, and therefore that patient would still not have an escape variant proliferating in their blood. We have two antibodies. We have our lead, our favorite antibody, and a second one that binds in a slightly different epitope, so they both can bind at the same time. So they're synergistic. The challenge with doing that is that it increases the cost of the medicine because you're having to do GMP manufacture on two antibodies and rather than one. And we're budgeting about $8 million for the GMP of our primary lead. We would need to come up with another $8 million to mobilize that second antibody. And that is a conversation we're talking with the FDA, and we're saying, look, if you're willing to pay for this, we can produce that molecule, that medicine that has the two. The other way to solve this problem is to find what's called a broadly neutralizing antibody, which is an antibody that binds a site that just the virus is unable to change. And that's a pretty effective technique for flu and for HIV because we've seen thousands of examples of flu and HIV. But that's not true here. We just don't know enough about where the mutations in nature is allowing this thing to diversify exists. There's some studies that people have done in petri dishes and test tubes, but it's not the same thing as understanding the evolutionary paths that are available to this virus. And so I like that concept, but right now that's not practical. So those are the approaches.
Got it. Got it. And now a question that many people had asked me when I was talking about antibodies, I was talking about you. And these are rumors, there are myths out there. There are some folks who are comfortable with Gates Foundation, some who are not comfortable. Again, I don't want to take a position one way or the other. My question to you is there any, what are the roles of the investors for this work that you are doing? Are you influenced by any external organizations? Positive influence, what kind of, what is your guidelight at this time?
I wish the Gates Foundation would give me money for this project because they have not done that yet. They've given me money for the flu project and I really appreciate it. So I know that there are these concerns that the Gates Foundation is like secretly an evil organization. I find it kind of confusing because there's so much money that they're spending to try to help the world. Like there are a lot of billionaires out there that really aren't spending much money at all to try to help. So I feel like if anything, we should be annoyed at those billionaires. No, I like what the Gates Foundation is doing. I believe in their message. I wish they would give me money for my COVID project. I'm confused why they haven't done that yet. So sorry to pick on you guys, but you guys should give me money. In terms of investors, I don't have venture capital investors. I do feel that government should pay for this. So I've been pushing hard to try to receive funding from government agencies. I appreciate the support of the Department of Defense, US AMRID, but that money so far just pays for some of the early stages. So we are having conversations with BARDA and we're hoping to receive their funding. That would be great because that would pay for our clinical studies. If I don't receive enough money for that, then I do need to go talk to venture capitalists and investors. And venture capital enables lots of medicine, but the reason I've waited for that to be my last choice is that venture capitalists, they're running a business. They want to make money. And so they're going to, if they come in and I bring them in, they're going to say, okay, well, I'll give you my money, but I don't want you to charge just $400. I want you to charge $3,000, right? So I've been resistant to that because I feel like I would rather solve a greater global good here and make some money than try to feel like I could make 20% or 80% more money, but then we're still stuck in this crisis. And ultimately, this is a global problem and we need medicine for everyone around the world for us to defeat this. And that is the balance. So if I could find the right investor who'd be okay with that, then I'd accept that. But I think they're right to ask, are there going to be influences from the money? And the answer is usually yes, which is why I really think government should pay for this because governments are paying so much money already because of their broken economies. Paying a tiny drop in the bucket to fix their economies by creating effective medicine seems like a no-brainer to me.
Absolutely. And it also seems like to me that if we can have GoFundMe to have a wall built and we can collect $25 million, I think the GoFundMe like things should actually put the money together for the companies like you. So thanks a lot for all of this great information. If you have a few more minutes, I would like to go over some of the comments. And if there are some questions there, I can ask you. We are 45 minutes into the talk. So would you be able to give us some more minutes?
Sure, let's do it.
Thank you. So there is, of course, a comment here, Jacob needs GoFundMe. So there is a question here. Antibodies decline after 3 months with SARS. If she wants to know if her immune system remembers best, try a T cell. So there has been a discussion about the T cells as well. That T cells and then the innate arm cells can also become memory cells and they can help in the long term as well. Is this more important to strengthen those systems compared to having these therapies, or they go hand in hand, or the therapy of monoclonal antibodies is superior?
So T cells, so antibodies are soluble factors secreted by B cells that go and bind up the virus. T cells are populations also from the adaptive immune system of an army of cells that have specific receptors that go detect if a cell is infected and they will kill that cell. They're also very important. There's two things about the antibodies. One is there was a bunch of news that came out saying that we lose our antibodies after 3 months with the novel coronavirus. It's just not true. If you go look at that study, first off, it was rebuffed by a study from Mount Sinai. And second, if you look at that specific study, look at the data, you'll see they did this, it's called R-plot. trend line. They had this trend line where there was just like a couple rogue data points at the end that didn't really even show that much of a die-off, and then it made the news saying there was a big die-off, but it's inaccurate. Go look at the primary data. You're fine. You're going to have the antibodies you got after being sick are going to last you at least 6 months, potentially out beyond a year. It is true that coronaviruses cause you to lose some titer over time, but it's not 3 months. It's probably going to be a year or 2 years. You're going to enjoy protection. That said, T cells are good, and you want T cells. The vaccines that are being produced right now, all three of them are eliciting T cell responses, and the reason that they're able to do that is that these vaccines are either pseudovirions, so they're like adenovirus or basically it's a virus that isn't COVID-19, the SARS-CoV-2, and then they put the spikes from SARS-CoV-2 on a different type of like less dangerous virus. So that does go infect your cells. T cells come over there and they learn how to chop those cells up, and so that does train the T cells as well as the B cells. And then the RNA vaccines from Moderna, the RNA gets inside of cells, so your immune system thinks that those cells are infected because they're producing lateral proteins. So those vaccines are producing both antibodies as well as good T cell responses, and that's a good thing. Those are both important overlapping layers of protection that you would want.
Got it. So there's a question here, which is, if a person already has antibodies, maybe they were infected before or they did not even know, if they already have antibodies, will injecting more antibodies cause any issue?
So no, it should be fine, and it may actually help, or it may be useless, but you're fine no matter what. So if you already have good antibodies, I know there's a lot of concern about whether or not that's good enough to protect you. I will say, as an immunologist, the smart money right now is that if you have antibodies after you've been sick, that you likely have immunity against this for a while. You're probably protected. That's just based on evidence of what we see with antibodies providing protection in non-human organisms like primates and mice and rats and ferrets, and it's also based on what we're observing with recovered patients. There are rare cases of people that seem like they get infected a second time, but the vast majority of people, we don't seem to see that effect. With respect to if you have antibodies, will more antibodies hurt? It shouldn't hurt. They're either going to show up and provide additional protection, which you may not need because your antibodies were already good enough. Or in the case of my antibodies, it might help because we've turned off those effector functions. So our antibodies, if you needed more antibodies, we could put our antibodies in. They will flood over the virus in a blanket that is silent to the immune system, which means you'll neutralize the virus without getting that inflammation reaction. And that could be helpful. I would say in general, if you have antibodies, you're probably okay right now. That's the current assumption.
Got it. And one final question, and then once again we'll say thank you for your time. The final question is the reinfection. So I don't know if it is directly related to antibodies or not. Is reinfection possible? Have you seen reinfections? Is it possible that the virus can attack again?
Yeah. So this has been an area of contentious data review. Here's where I think we are, and again, the data is incomplete. I think there are some cases that look like reinfection took place. Initially, there was reports about this being pretty common in South Korea, and then they realized that what was happening was that the assays, the kits to detect whether or not the person had the virus were not very reliable. And so they would test someone repeatedly, and they were like, oh, it seems like the virus went away and then it came back again. I think it just never actually cleared, and for a while they failed to detect. There have been some other cases of pretty well documented people who completely cleared and they got it again. And we also know that there's this very long period of occult infection where you're infected and you don't show symptoms. So it's an odd virus in the way it sort of has a long fuse inside of the person. We also know that there's a wide range of people who get infected, and they have from extremely good antibodies to absolutely no antibodies. The data is not as well studied for T cells, but it's probably the same thing. And so I feel it is possible that you'd have some people who got infected, recovered, and they just didn't mount a decent immune response. And that means that they were vulnerable to be infected a second time. We do know that the ACE2 receptor interaction of the RBD is extremely high affinity. So that means that this thing binds very tightly to those ACE2s. And we observed that in our hamsters, that in the prophylactic model, we dropped the amount of infection by 10,000-fold by giving the antibody up front, but it didn't entirely block it from getting in there. It just really reduced it. And that's just because this thing grabs on like Velcro so tight. we can probably stop it from spreading, but it might get in there a little bit. And so if the person had a weak immune response, it's possible they could get reinfected. So that said, we think it's possible, but I think it's very rare. There's a limited number of cases that have been observed over millions of people who've been infected. So I still think the most likely scenario is that someone who's been infected and has recovered, provided they have good antibodies and or good T cells, they probably aren't going to get infected again. And the best way we're going to find this is by looking at looking at doctors and nurses, because those people are constantly being exposed to this. And so you look at the ones who've recovered and they've returned to work and ask how often are those people infected a second time.
Got it. Last question, and I think this is a brilliant question. Last question is, do you think that these antibodies can cross the blood-brain barrier or placental barrier, or can they be secreted in the milk from the lactating mothers, and what is the effect of that?
That's an awesome question. So in the body, normally with normal antibodies, breastfeeding mothers will create a type of antibody called IgA, and that antibody is specially designed to work in mucosal membranes, in particular to cross the breastfeeding. And the infant's stomach is not very acidic, and it's set up to be able to take those antibodies and internalize them. So that's one of the ways that a mother is able to protect their child through breastfeeding. The antibodies that I've engineered and other groups are engineered, those are IgG, so they're not really designed to go do that process, which means the baby would need its own medicine. With respect to crossing the blood-brain barrier, the blood-brain barrier is imperfect in its ability to block antibodies. We see typically antibodies, about 1% of antibodies in the peripheral blood are making it across the blood-brain barrier. So they get across the barrier, but just not very efficiently. That gets more efficient in cases like people with multiple sclerosis who have other diseases of the blood-brain barrier, or if there's blood-brain barrier inflammation. But 1%, that's a problem. So if you actually needed this drug to work across the blood-brain barrier, if you have 1% of the dose in there, it's probably not enough to be effective. Fortunately, it seems here that the vast majority of the disease process is happening in the periphery, because the blood-brain barrier also blocks virus from crossing. That's part of its purpose, is to create a castle wall around the really important parts of the neurological functions. But to your point, no, they don't. The current antibodies don't. There are ways to engineer antibodies to cross the blood-brain barrier, hitchhiking across the transferrin receptor, but I don't think anybody that I know of is applying those technologies to these therapies.
Got it. And with every question I say, this is my last question. Now I have one more question, which is a brilliant question, and hopefully this is the last one. The question is, those folks, for example, seniors who have a weak immune system, probably vaccines are not going to make them respond that well and make antibodies. So in these folks, number one, is it easy for them to become reinfected? And number two, is it important for them to have these antibodies available so they can be protected?
Yeah, so the elderly have something called immunosenescence, and that's just a fancy way of saying that the immune system gets old when you get old. And so the cells are they don't activate as easily. It's kind of all the stuff that old people do. They want to get off the couch. They're like, oh, everything hurts. The immune system is doing the same thing. And that means that the elderly don't respond as well to vaccines. And that's a problem, because the elderly are most likely to have very severe reactions to the coronavirus infection. For that group, still, if you're elderly, you should still get the vaccine. Wait for the studies to show that it's safe in the elderly to make sure that the reactions aren't more severe, but I would still take it. But for a large number of the elderly and for the immunocompromised, there are people that are either they have cancer, so they're receiving medicines that are knocking out their immune system, or they just have a genetic defect that's causing them not to produce a normal immune response. For those groups of individuals, they should definitely appreciate having an antibody therapy, because the antibody works the same and everybody who receives it. It works immediately, and it'll give them a couple months of protection. And that would be a strategy that I would recommend for that cohort, that they receive an antibody therapy every couple of months until the incidence of the virus is lowered through herd immunity from the larger population.
Awesome. So thank you very much once again, Dr. Glanville. It was wonderful to have you with us. One thing that I'm very proud of is the cool beans here, the viewers here. We have been for now about 110 days. We have been going over various parts of the immune system. We understand how the antibodies are, how the T responses, B responses are. So this was a great additional view on top of it. And we were always very curious about the antibodies and their usage. So thank you very much for coming in and answering those questions.
Thank you very much for having me on, and thank you all, cool beans, for your attention.
Thank you. Bye-bye for now.
Cheers.